Hello, today we will have a quick look at AgentD, a computational framework that utilizes large language models to automate and enhance early-stage drug discovery. By integrating LLM reasoning with specialized tools, the agent handles essential tasks such as data extraction, molecular generation, and property prediction. A standout feature is its property-aware refinement process, which uses targeted structural modifications to improve the safety and efficacy of the candidate molecules. The framework also employs retrieval augmented generation to provide literature-grounded answers to domain-specific queries. Comparisons across different models show that while performance varies, the Agentic workflow consistently improves drug likeness and provides detailed mechanistic insights. AgentD coordinates 6 essential modular tasks by integrating LLM-based reasoning with domain-specific tools and databases. The system is compatible with LLMs from OpenAI, Anthropic, and DeepSeek, although its primary development and execution environment relies on OpenAI models. Yet, DeepSeek was found to be equally efficient. The pipeline begins with the data extraction stage. In this phase, AgentD retrieves critical information from both structured databases and unstructured web sources. For a given protein disease context, it identifies known drugs when available and collects protein FASTA sequences from UniProt as well as SMILES, representations of known compounds from the Chem database. Additionally, the agent autonomously constructs keyword-based search queries to retrieve relevant open-access scientific literature from Semantic Scholar, providing broader scientific context for downstream decision-making. To address domain-specific scientific questions, AgentD employs a retrieval-augmented generation strategy. Next, the SMILES generation module produces candidate molecules. AgentD can generate seed molecules using external generative models through 2 complementary strategies. The first uses reinvent, which enables de novo molecule generation without requiring starting compounds. The second employs Multimo, which performs conditional generation to create structural analogs of a known molecule, such as a drug identified during the data extraction phase. Once candidate molecules are generated, the property prediction module evaluates their pharmacological potential by estimating key drug-relevant properties. More than 60 properties related to absorption, distribution, metabolism, excretion, and toxicity are predicted using the DeepPK API. In addition, binding affinity is estimated using BOP-ULM, which analyzes both the ligand's SMILES representation and the protein's FASTA sequence. The molecule refinement module then improves candidate structures based on the predicted properties. The agent identifies molecular weaknesses such as high toxicity or low permeability and proposes targeted structural modifications. Unlike the other modules, this refinement step relies entirely on the LLM's internal reasoning and embedded chemical knowledge rather than external tools. Finally, the structure generation module produces 3-dimensional protein ligand complexes. In this stage, AgentD uses Boltz-2 to generate 3D structures from the ligand and the protein sequence. The resulting models provide estimated binding metrics, which serve as preliminary indicators for prioritizing candidate compounds. Here is an overview of the different steps. The process begins with a user-provided query, such as identifying drug molecules that target a disease-associated protein. The AgentD framework first determines whether known drug molecules exist for the specified target or disease. When available, these molecules serve as starting points for subsequent molecular exploration. Because AgentD also includes a de novo generation module, the workflow can proceed even when no active molecules are identified. In such cases, the SMILES generation task constructs a chemically diverse library of candidate molecules using generative models. The generated molecules are then evaluated using property prediction tools to estimate key properties, including predicted binding affinity values. No 3D structures are required at this stage. Based on these predicted properties, AgentD proposes SMILES modifications to improve attributes such as solubility or metabolic stability. These refinements rely on the language model's internal reasoning rather than external optimization tools. Following refinement, domain-specific criteria defined by the user can be applied to select promising compounds. Throughout the workflow, users may also request clarification of scientific concepts. AgentD was evaluated on 3 therapeutic targets, and the results showed that after a few rounds of refinement, the number of molecules with a quantitative estimate of drug likeness score above 0.6 increased substantially. That is, the model selects or generates better candidates step by step. Similarly, the number of molecules satisfying other types of empirical drug likeness scores also increased. That is it for today. I hope this short introduction to AgentD gave you a helpful overview of how it can support your research. If you found it useful, feel free to explore the project further. I've included the GitHub link in the description below. Thanks for watching, and until next time, take care.